The present video is about our ongoing research project on the ancient genomics and the molecular mechanisms of human tolerance to arsenic in northern Chile. The Atacama Desert, the driest place in the world, is located in northern Chile. This has been an extreme environment for human survival due to high UV radiation, hypoxia at high elevation, and toxic levels of arsenic in rivers and wells. However, first human populations, such as the Chinchorro culture, who colonized the coastal environments and valleys, such as the Camarones Valley, relied mainly on marine resources from the Pacific Ocean. The formative period started about 3,800 years before present, and this is characterized by the introduction of agriculture and the extensive use of domesticated animals. In addition, there is an increased number of inland archaeological sites along the valleys. The intermediate and late periods in this valley are characterized by higher interaction with other societies in the Andean highlands, such as the Tiwanaku culture and the Inca. Our project is centered on understanding the cultural and biological diversity of pre-Hispanic and present-day populations that have been living in environments with higher levels of arsenic in northern Chile. Particularly, we are interested in answering three main questions. First, what is the genetic relationship and history of the ancient and modern populations in northern Chile? To answer our first question, we will reconstruct the genetic history of coastal populations from northern Chile by using ancient and modern DNA samples. Specifically, the ancient DNA samples correspond to archaeological tooth samples that have been collected at the National Museum of Natural History in Santiago, Chile. In the ancient DNA laboratory at the Arizona State University, we will extract the DNA and use it to make DNA libraries and sequence them. After getting the sequences, we will analyze the data by comparing the reconstructed ancient genomes with data from modern Andean populations. Our second question is, how has the agricultural transition impacted the oral microbiomes in these pre-Hispanic populations? To answer this question, we will characterize the ancient oral microbiomes from pre-Hispanic populations that had two different diets. For example, the ancient Chinchorro people had a marine-rich diet, while later agricultural populations had a plant-rich diet. Our third question is related to the evolution of arsenic tolerance in northern Chile. How do the mutations associated with arsenic tolerance affect the expression of the AS3MT gene? Over thousands of years, Andean people in the Camarones Valley in northern Chile have been exposed to naturally high levels of arsenic from the Camarones River. Indeed, when people from the Camarones Valley drink this water, they can metabolize the arsenic very quickly and excrete it through urination. The main enzyme responsible for this metabolic process is the arsenic 3-methyltransferase enzyme, or AS3MT. Thus, we have been studying the genetic basis of this capacity in the Camarones people by analyzing their unique mutations in the AS3MT gene.